From a drop of insulin in a petri dish to the footprints of mammoths on frozen ground, our mastery over biology has taken us to incredible frontiers. Once, biotechnology saved lives by reprogramming bacteria. Today, it promises to bring back what was lost. This is the story of how the tools of life sciences have evolved from healing our bodies to rewriting history. The roots of biotechnology go back to fermentation and the accidental discovery of antibiotics. But the modern era began in 1978, when scientists at Genentech inserted a human gene into bacteria. The result? Synthetic human insulin, the first drug produced through recombinant DNA. Before this, diabetics relied on insulin from pigs and cows, less efficient and sometimes dangerous. But this breakthrough made insulin cleaner, safer, and widely available. It was the first time we used biology not just to observe life, but to redesign it. Biotech quickly spread. Vaccines, cancer treatments, biofuels, and GMOs, all products of our growing ability to manipulate DNA. Biotechnology didn't stop at medicine. In agriculture, it reshaped how we feed the planet. Genetically modified crops like Bt corn and Roundup Ready soybeans boosted yields and reduced pesticide use. In medicine, gene therapies now allow doctors to treat rare genetic diseases at the source with DNA. One of the most game-changing tools, CRISPR, precise gene editing technology likened to molecular scissors. With it, scientists can fix mutations, engineer drought-resistant crops, or even edit human embryos, sparking debates about what it means to be human. Today, the biotech industry is worth over $1 trillion and growing. From pandemic vaccines to lab-grown meat, its impact reaches every continent. Now, a new chapter begins, using biotechnology not to save life but to revive it. This is de-extinction, the process of bringing back extinct species using advanced genetics. Colossal Biosciences, a Texas-based company, is leading the charge. Their goal? To bring the woolly mammoth back to life by editing the genome of the Asian elephant. Using CRISPR, scientists are inserting mammoth genes like those for cold-resistant hemoglobin and thick hair into living elephant DNA. The result? A hybrid designed to survive an Arctic tundra. And they're not stopping there. The dodo bird, the Tasmanian tiger, and even the giant ground sloth are on the wish list. Once science fiction, de-extinction is fast becoming reality. But just because we can, should we? Critics warn that de-extinction could distract from preserving species still alive. Reintroducing long-gone creatures into modern ecosystems could trigger unpredictable ripple effects. What happens if a revived mammoth tramples Arctic vegetation needed by today's caribou? There's also the question of suffering. Would cloned or engineered animals be healthy? Would they suffer from deformities or social isolation? On the other hand, advocates argue that these species were often driven to extinction by humans and we owe them a second chance. Mammoths, for instance, could help slow climate change by knocking down trees and keeping the tundra frozen. As Harvard geneticist George Church puts it, de-extinction is not just about the past, it's about the future of ecosystems. We stand at the threshold of something enormous. In the coming decades, biotechnology may eliminate inherited diseases, grow human organs, and even redefine aging. With de-extinction, we gain not only ancient animals, but ancient knowledge of how genes shaped evolution, of how ecosystems can be repaired. But these powers come with moral weight. We're not just rewriting genes, we're rewriting stories, histories, and the balance of nature. The future of biotechnology is just scientific. It's political, environmental, and deeply human. What kind of world do we want to create and who gets to decide? Just as the printing press once turned knowledge into a public good, the tools of biotechnology are now passing from the few to the many. By the early 21st century, the genome, the once secret script of life, had become legible not only to elite scientists, but to classrooms, grassroots labs, and remote communities across the globe. From high school students in Kenya designing microbes to clean rivers, to indigenous researchers in Alaska merging genetic science with traditional ecological stewardship, a new chapter was being written not in ivory towers, but at kitchen tables and field stations. This was the democratization of the gene, a movement as historic as it was hopeful, where the power to alter life itself became part of a shared human inheritance. History has always been marked by the stories we leave behind etched in stone, encoded in scrolls, whispered through generations. But in the early decades of the 21st century, a new kind of legacy emerged, one not written in ink or stone, but in the double helix of DNA. For the first time in human history, we could not only record our past but revise it. Species lost to time could be recovered, inherited diseases rewritten. Even evolution itself, once blind, could be steered by human intention. Historians may one day look back on this era as a biological renaissance, an age in which the blueprint of life passed from the realm of fate to the realm of choice. But they will also ask, what did we choose to preserve? What did we choose to create? And what, in the end, did we leave behind? The future isn't written in stone, it's written in code, DNA code.